Right here, we have diffusion. What is diffusion? Well, if you look over here, if we go back to the cell, we can see that the cell doesn't have a door or doesn't have a mouth, right? So how do we get the material that's necessary inside the cell? It's gonna be obtained or released by the cell by a process called cell transportation. Since the transportation happens through the cell membrane, then this is also known as the membrane transportation. So then what are we talking about here? Well, we said, for example, that we need to create ATP, right? So then this mitochondria produces ATP. Okay, good. And what is it that mitochondria, how is it that mitochondria is able to produce ATP? It's not going to do it through magic, right? So then that means that something must go inside here, and then something will come out in the form out of the cell in the form of ATP. So then what goes in here? Well, in there, you're going to have to put in there oxygen. What else you need to put in the mitochondria? Well, you have to put in here, for example, carbohydrates, like a piece of bread that you ate this morning. So carbohydrates will go in there. And what will come out of the cell? Well, ATP. What else? You're going to get also carbon dioxide. And you're going to get out of here also, you're going to get in there water, H2O. So as you can see here, there are things that need to go inside and outside the cell. What is the name of the process by which these things go in and outside the cell? That's going to be called cell transportation or membrane transportation. How do this process of cell transportation happen? Well, it's going to happen in two different ways. One of them is going to be active transportation, and the other one is going to be passive transportation. Right? Active transportation uses ATP, right? And passive transportation doesn't use ATP, it's going to use difference in forces, right? What does that mean, difference in forces? Always from high to low. What are we talking about from high to low? Well, we said that, for example, in our body, we have our blood, and we can say, therefore, that our blood is a solution because you're going to have solutes and solvents, right? Remember, we are talking about the human body. That's why I'm talking about the blood, right? So then solutes, it can be what do you have in your blood. You're going to have, for example, sodium. Right? You're going to have solvents such as water. So then passive transportation, if you use sodium, for example, when we talk about solutes, we're going to, example of that is going to be diffusion. If you talk about water, example of that is going to be osmosis. So therefore, how come passive transportation happens? Always from high to low. High to low what? For example, if we talk about sodiums, high to low, right? So then that high what? High, if you talk about forces, right? If you talk about sodium, you're going to talk about concentration. If you talk about solvents like water, then you're going to use in here pressure. So when we said high to low, high to low what? High to low concentration, if we talk about sodium such as sodium. Or high to low what? Well, high to low pressure, and we are talking about water in the case of solvents. Examples of high to low, when we talk about high concentration, then we are talking about diffusion. An example of high to low, when we talk about pressure, it's going to be osmosis. Or filtration, for example. Okay, so then if we scroll down right here, what are we talking about right here? Well, we are talking about diffusion. So if we talk about diffusion, then we are talking about solutes. I remember I told you, solvents, pressure, right? Solutes, concentration, right? So therefore, example of things moving from outside the cell, inside the cell, because it goes from high to low, solutes, then that's going to be diffusion. Okay, this is cell membrane right here. That's the cell membrane. And if you look over here, we say high to low, right? High to low. So let's take a look at here. Then, I don't know, let's say you have in here, let's say these red tomatoes. Okay, you have a whole bunch of red tomatoes in here. You have no red tomatoes in here. And we said it always moves from high to low. And that's why you see how the tomatoes, right, after a while, start moving over here because it goes from high to low. And this is an example of diffusion. We can talk about what factors are going to affect this diffusion. So if you really think about it, you have these red tomatoes. Imagine that you have, green tomatoes right there. And you have only 3 green tomatoes. Which one do you think is going to go through first? Well, the red tomatoes. Why? Because there's more. So therefore, you can say that quantity affects how diffusion happens. Imagine that you have something else, but this These other things are this big right there. So then which one do you think is going to go first through the cell membrane? The red tomatoes that are tiny or this big thing right there? Cell tomatoes that are tiny. So therefore, you have to agree that the size of the molecule also affects how these things go through it. So imagine that this membrane is actually made of bricks. Do you think that the red tomatoes will go through it? Obviously not. So it doesn't matter what you have on the other side, nothing will go through it because the cell membrane also affects how these molecules are going to move through it. So therefore, the characteristics of the cell membrane also affect that. Temperature also affects how these molecules move through the cell membrane. As you know, when you have an ice cube, what happens? The molecules are completely frozen in there, right? They don't move. Now, imagine that you put water and you put it in your oven and the water is boiling. Water molecules are going to move, right? They're going to move a lot. So then which ones do you think are going to go through diffusion first? So obviously the molecules that are going to be in higher temperature, because you can see in there the molecules of water move way faster when the water is boiling when compared to the ice cube. So therefore, temperature also affects how diffusion happens. That's going to be diffusion. So then if we scroll down right here, now we need to talk about osmosis and tonicity. In this case, well, tonicity is how the solution affects the water that we have. Okay, so let me show you that in here. This is a membrane, right? This one right here says phospholipid bilayer. That's the membrane. So we're saying that this is the membrane like this. So then we can make this go like this. And we can say that this is the nucleus right here. So therefore, this is going to be outside the membrane, and this is going to be inside the membrane. And inside the membrane means inside the cell. So then as you can see here, guys, the amount of red tomatoes, right? This is outside, right? This is outside, and this is inside the cell. So you can see here that the red tomatoes, right, and everything that is going to be outside is going to depend on the amount of red tomatoes that you have inside. So let's say, for example, this is 1 minute, this is 5 minutes, and this is 10 minutes. Okay, so then you can see here that after 1 minute, nothing happens. So right here, because in 1 minute, this is the amount of tomatoes that we got in there after 1 minute. But then what happens after 5 minutes? As I mentioned before, the tomatoes start going in here. And the cell starts getting tomatoes inside. Now, is there something that the cell can do about these tomatoes? Imagine that the cell doesn't want the tomatoes. Can the cell do something about it, about these tomatoes? No, because this is passive transportation. It just happens from high to low. So therefore, do you think that the tomatoes outside affects what's going on inside the cell? What do you guys think? Yes, the tomatoes outside, right, have an effect on what's going on inside the cell. Whatever happens outside the cell will affect with what happens with the things that happen inside the cell, right? So then whether the cell wants that or not, these things will go inside because high to low, right? In this case, concentration, for example. Not only that, not only the red tomatoes will go in there, but the amount of water that you have in here, the blue will go in here too, depending on the amount of water that you have. Imagine that this is your blood, guys. Imagine this is your blood. So then this is a red blood cell. Okay. Now, is your blood a fluid? Yes, your blood is a fluid. How come your blood is a fluid? Because it has water. Okay, so therefore that means that this in here is going to be water. What else do you have in there? Don't we have gases, for example, oxygen and carbon dioxide? Don't we have nutrients like the salt that you eat with your French fries? Yes, right. So you have all that. So then right here you have a fluid. You have solutes in here, such as, for example, as we mentioned before, is salt. Right? You will have all those things. So therefore, as we mentioned before, what happens inside the cell depends on what you have outside the cell. And since we have water, which is a solvent, and you have salt, which is a solute, then this outside the cell is a solution. So you know that the solution outside will affect what's going on with the cell inside. So how the solution affects what's going on with the cell, that's going to be called tonicity. So the tonicity is how the stuff that is outside in this solution affects what's going on with the cell. Okay, so then what's tonicity? Tonicity basically is how this stuff that is outside, how this stuff, the tomatoes and the water, how that affects what's going on inside your cell. And you know, based on what we talked, that that's going to happen no matter what. So then this is the cell membrane. Okay, and we say we have homeostasis, right? We say homeostasis, right? So let's say, guys, that we have in here, I don't know, let's say 30 tomatoes. And in here we have 30 tomatoes. And we have 2 liters of water. And in here we have 2 liters of water. So this is equal, right? So then when this solution matches with what you have inside, then this type of solution, remember, the solution is what you have outside, right? So then when that matches with what you have inside the cell, then this type of solution is going to be called isotonic, right? Isotonic. The other one is going to be, for example, hypertonic. Hyper means too much, right? This is high. So then that means that in this example, right, we don't have 30. We have, I don't know, let's say we have 40. Let's make it 80 to use this thing. So this is hyper because outside is more than what we have inside. So therefore it's going to be hyper. Now, what's the other possible option? Then the other possible option is going to be hypotonic, in which this means that outside is going to be less, right? So in this case, what we're going to have in here, 10, and inside is going to be 30, right? So therefore, the only case in which the cell is okay is when it's going to be isotonic because the solution is going to be the same. Now, when it's hypertonic or hypotonic, then that is going to affect what things go in or what things go out, because the solution in here will affect, as we agree, with what's going on with your cell. So then if we look over here, that's why it says in here that we're going to have isotonic, hypotonic, and hypertonic, depending on what the solution is going to be outside, right? So you're having here more or less the same thing that we just said, the tomatoes and the water, which I explained already in the other figure. So if you look over here, you have, as I mentioned before, the example that we have right there with the cell. So right here, basically what it says is that this one right here is going to be isotonic. So right here we have 3 sodiums inside the cell, and right here we're going to have 3 sodiums outside the cell. So then what happens with the water that we have? Nothing, because since they are the same, this is exactly the same, so nothing happens with the water. So you can see here then that your red blood cell, when it's isotonic, it remains with that very beautiful shape that it has. Now, this is going to be A, as you can see here, isotonic. So then what happens in hypotonic? So we said that inside the cell is going to be 3. Inside the cell, outside the cell, the solution is going to be hypo. So then that means if this is going to be hypo, this is going to have to be less. So let's say this is going to be 1. So when you have less in there, the water that was outside will go inside the cell. And because of that, since the water will go from outside to inside the cell, you can see here how the cell is inflating, is ballooning, right? It's going to become so large that eventually the cell will explode. The other situation in here is this one right here, when we said we're going to have hypertonic. So in that case, the cell is going to be 3. If this is hyper, it's because outside is going to be more. So let's say this is going to be 5. So then in that case, what's going to happen with the water? The water from the cell will try to go out in order to balance this. And because of that, the cell is going to start losing a lot of water. But you can see how it shrinks in here like this. It's becoming smaller and smaller because obviously the cell is actually losing water. This one right here is called crenation, right? So then when the cell shrinks.